Good afternoon, everybody. It's 12 p.m. here in Toronto. Please mute your microphones due to high number of participants. We're back with our first inaugural series of molecular pathology for surgical pathologists. And today, Dr. Yael Fisher will continue to present. Yael, please, the floor is yours.
Thank you, Anna. So today this is our third meeting and we're going to talk about bioinformatics. I still have nothing to disclose. So I want to start with the definition. What is bioinformatics? It's a subdiscipline of science that combines biology, computer science, and mathematics to collect, store, and analyze and disseminate biological data and information. This information is mostly DNA and amino acid sequences or annotations about these sequences, and they're organized and indexed in databases that are mostly publicly available. Cancer bioinformatics refers to identification of alterations that are associated with cancer in order to understand the pathogenesis, identify therapeutic targets, follow the course of disease, and in total, improve patient outcome. But we're pathologists, so why do we care about these bioinformatics? So in the past 2 decades, molecular techniques were introduced into the clinical practice in pathology. It started with identification of specific known mutations, usually using PCR or Sanger sequencing, and it gradually grew into larger and larger panels. Interpretation of variants in known hotspots is pretty trivial. We know the variant, we know what it means, but the introduction of NGS has presented with new challenges. So NGS is massive parallel sequencing, meaning we're sequencing multiple targets at the same time. In large panels, this can be thousands of targets with millions of sequences as a result. Now, some of these result in variants that are known hotspots that, again, we know what to do with those. But others are rare variants or novel variants that we don't really know what to do with them. And our job as pathologists is to integrate the morphology and the results of the sequencing together with everything that is known about these variants into our molecular or into our pathology report. So we started the series talking about the basics of molecular biology, the central dogma of molecular biology, where DNA is transcribed into RNA, that's translated into protein. And we talked a little bit about the structure of DNA and RNA. We continued with talking about the different types of variants in the genome, and most of our focus was and is going to continue to be on point mutations, including single or multiple nucleotide variations or short insertions or deletions. We also talked a little bit about the nomenclature and how to read the name of the variant that we are seeing. In our second meeting, we talked about the laboratory side of molecular pathology, starting with selection of an adequate specimen for testing, continuing with extraction of DNA. We talked about the importance of quantification and quality assessment of this DNA we have, and the different methods of sequencing. We talked about PCR that really started this whole thing. Continued with Sanger sequencing that used fluorescently labeled nucleotides that terminate the replication of DNA and are then read into a complete sequence. And we talked about about NGS, starting with the different approaches to build a library, the amplicon-based approach using primers for each end of the sequence we're interested in, or the hybridization capture-based approach that uses probes to capture the regions of interest. And we finished last time with talking about the different methods of sequencing in Illumina and in Ion Torrent. And of course, there are other methods as well. At the end of the session today, hopefully you'll be able to recognize the different types of output files and describe their purpose, summarize the bioinformatics pipeline, utilize IGV to visualize and verify variants, recognize some common sequencing artifacts, and understand the rationale behind using a tier-based variant classification scheme. So again, we're talking about next generation sequencing, and this is a summary of the whole process from tissue to a list of variants to analyze. So we start with extraction of DNA, library, continue with library preparation, and then sequencing. And at this point, we stopped last time. And today, we're going to continue this sequence from the results of sequencing until we have a list of variants to analyze. And hopefully next time we'll talk about the interpretation of these variants. So the first type of file I want to introduce is the FASTA file. This is a text-based file. The ending of the file is .fasta, but it can be open in any text viewer, like even in Notepad. This file contains 2 rows. The first one starts with this greater than sign, and then there's a header, the name of the file or the sequence. And it continues with a sequence that contains the 4 nucleotides A, C, T, and G. It can contain any sequence, a gene, a chromosome, or even the whole genome. And it's used as a reference file to identify variants later on. So these are some examples of a few FASTA files. These 3 are all FASTA files of chromosome 1, but each of them is from a different assembly. Okay, so the first one is GRCH37, then we have GRCH38, and then this file is from the newest assembly, the T2T assembly. Now, if you notice, these 2 actually have the same excision number, just a different version. So they're not that different. While here in the T2T assembly, there's a completely different excision number. So this sequence is really different from the others. These 2 start with a long sequence of Ns. N means that the nucleotide is not identified. And these in GRCH37 and 38, the full sequence of the telomeres was not available yet. But if you scroll down in these files, you'll eventually get to a spot where you can see the actual sequence. In the T2T assembly, you can see the actual sequence right from the beginning. And this is another example of a FASTA file that is of that shows the sequence of the KRAS gene. You can see the excision number at the beginning. Each gene can have different excision numbers, so it's good to know what we're looking at. Now, if I go back to the process of NGS, the first step in this process is the base calling step. And this means that we're taking our raw data that can be an optic result or an electronical map, depending on which type of sequencing you're using, and turning it into a file that contains the DNA sequence. And now, if you can see, this is similar to the FASTA file, but it contains a little bit more data. And this is called the FASTQ file. FASTQ stores the sequencing data together with quality scores. Each read in the FASTQ file will have 4 rows. The first row starts with an @ sign, and then there is the read name, which is usually assigned electronically by the sequencer. Then it contains our sequence. The third line starts with a plus, and an optional description can be added. There are usually, in most sequencers that I know, it's kept empty. And the fourth line is the quality line that corresponds to each letter in the sequence. So each of these letters get a different quality assignment. This is done by different characters that I do not know how to interpret by heart. But in the rest of the process, the algorithms can use this to interpret the quality of our findings. Now, each FASTQ file will contain millions of rows, okay? 4 rows for each read or each sequence that was produced by the sequencer. Now, this is great. We have our sequence, but no one can look at at millions of sequences and identify what went wrong with it. So to continue this process, we do a process that is called alignment. Alignment means arranging the sequencing results, our FASTQ file, with a reference sequence, a FASTA file, in order to identify alterations. Now, for our purposes, we're talking about clinical practice in molecular pathology, so the reads are usually aligned to a human genome reference, and we usually use the GRCH37 version, although occasionally you might see the GRCH38 version. Depending on the assay you use, sometimes for RNA, there are specialized reference files that contain the fusion sequences that assay tests for. In other assays, RNA is aligned to the human genome, just like DNA. So it really depends on which assay you use. The results of the alignment process go into a BAM file, that's a binary alignment map file. And these can be visualized in IGV. You've seen this view in our series, and I'm sure you've seen it in other places too, and I'll talk about it in more details just in a few slides. After we have a BAM file and we aligned our sequences with a reference sequence, we can go into a process that's called variant calling, which identifies the changes in our sequences. So this process is done by algorithms that are called variant callers. There are different algorithms that can do it, and in turn, each algorithm may have slightly different results. Okay, so sometimes when you don't see a specific variant you're looking for, using a different algorithm might help. And the results of this process of variant calling goes into a file called VCF file. Again, it's a text-based file. And let's talk a little bit about how it's built. So VCF is variant call format. And it has this table of columns. It starts with the number of chromosome where the variant is, the chromosomal position of the variant. Some variants may have a variant ID depending on what the variant caller uses for identification. So if it identifies a variant that it knows, it can assign an ID to it. Then we have the reference at that location and the alternative allele that was identified in our sequence. There's a quality score that is based on the quality we saw in in the FASTQ files. But this is more easy to assign the probability that this base call is correct or incorrect. Okay, so and it's a logarithmic scale, and this is how to interpret it. So if you have a score of 10, that's pretty low. It means that 1 in 10 might be incorrect. So I don't know, this variant with a score of 3, I'm not sure I would trust it. Okay, but variants that get a quality score of 67, that's above 50 here. So 1 in 100,000 chance to get an incorrect base call. That is probably a correct call here. If a filter is applied in the algorithm, you'll see a pass or fail score here. You can also see the read depth, okay, meaning how many reads we got for this specific sequence, and the allelic frequency where this variant was seen. So for this first variant here, the coverage is, or the depth of read is 14. We had 14 copies of this specific sequence, and this variant was seen in half of them. So that's a pretty good score for this. Now, this file is great. We can see our sequences, but I don't know what this means. No one remembers what position for 1400-370 on chromosome 20 is. I don't know what gene that is. I don't know if this is significant. So the next step in the process is to annotate these variants and assign functional information to what we had in the VCF files. So the coordinates we had are translated into genes, codons, and amino acid changes, and some bioinformatic data from different databases is added to the specified variants. Now, these annotated files can be different depending on the bioinformatic pipeline that you have. But this is just an example of how it might look like. So it starts with the chromosomal location and with the number of the chromosome and the position on the chromosome. And then we have the reference base and the alternative base, just like in the first VCF file we saw. But then the rest of the data is much more accessible. You have the allelic frequency of the variant, the depth of coverage. Here in the next column, and then you have the name of the variant in HGVS nomenclature. So you have the gene name, the base pair change, the excision number of the sequence that was used as reference, and the protein change here. You can get the variant type if it's a missense variant or a frameshift variant, and some information from external databases. So this is ClinVar database, and this is the Cosmic database, and even information from prediction tools that are more useful when interpreting germline variants, but may sometimes be useful in the somatic setup as well. So this file is something that is much easier to work with. And now we're getting to the step of analysis of these files, but we still have multiple variants in these files, and we need to know which ones we want to use and which ones we do not. So this process of analysis has two steps. First, we need to recognize whether the variants we're seeing are true variants or they might be artifacts as well. And then the second process is to interpret them and determine if they have any clinical significance. So today I'm going to talk more about the first part. And to do that, I'm going to talk a little bit about IGV. So IGV is an integrative genomics viewer. It's available for free download on their website. If you just Google IGV, you can download it. I think it's from Broad Institute. I'm going to talk a little bit about the basic features of IGV, but if anyone is interested in going more deep into it, this is a link to their user manual that is very user-friendly, and you can find out a lot about it. So let's talk a little bit about how IGV is built. This is the general window. It has many different parts that I will try to specify each one of them. I prefer to use the downloaded version of IGV, where you download it from the Broad Institute and put your BAM files into it. But depending on the software you use, you might have access to like a light IGV or an internet-based view of IGV that is basically the same. It has the same features. Maybe the colors are a bit different, but you can do everything you can do in the regular IGV in this too. So let's talk a little bit about how this is built, and I'm going to start with these three boxes here in the top left corner. So the first box is the genome assembly, and here in this case it gives us the GRCH37 assembly. But if you click on this arrow here, you can change the reference genome into any genome you'd like. So there are different versions of the human genomes are here. Okay, the HD19 or GRCH37. You have the 38 version, and also the T2T genome assembly. But there are different genomes here too. Now, I never thought I'd use them, but there happened to be a restoration project returning this subtype of gazelles to nature, and they approached us to help them with sequencing the DNA of this gazelle. And it turned out there was no good reference sequence for gazelles that was available, and the closest one was Bostaurus, aka the cow, and we use this reference sequence for these gazelles' DNA. So you never know what you'll come across, and this might be useful for someone else too. The next box here is the chromosome number, and you can also, again, click on it, and it will give you a view of the whole chromosome. If you want to know, where the genes are on the chromosome, or the order of the genes, sometimes that might be useful. For instance, when you're looking at translocations. Okay, so you can choose the chromosome of interest here. This will give you most of the genes, but the resolution would be too low to see the actual sequences, and in order to see them, you'll need to zoom in much more than the chromosome level. And the third box here is the search window, and here you can type in anything you want to search for. So if you type in the word all, you'll just get the whole reference genome here with the different, each number is a different chromosome, of course. You can type in the chromosome you're interested in. So this is CHR7, right? And then you'll get the exact same view that you got when choosing the specific chromosome you want. But more interest is to look at the variants that you're interested in. So this is an example that I randomly chose from one of our files. It's a variant in the MITF gene, causing a change from C in position 45 to T. And this is a synonymous variant, not changing the amino acid here. Now, if I want to search for this specific location in IGV, I can type in 3 different things into the search window. I can type in the chromosomal location. Even if you copy it from the Excel file and paste it into the search window, it works perfectly. Okay, so you can do that. You can type in the coding change. Okay, so you'll type in the name of the gene, a colon, and then the position where the change occurred and the change that occurred. And probably the most useful is to type in the amino acid change. Okay, so again, the name of the gene, a colon, and then the amino acid that changed in 1-letter code. So in this case, we have a synonymous variant, so it's H or histidine in position 15 to histidine. You always need to type in the last letter, okay, the change. If I do just H15, it won't work. But it doesn't really matter what you type in here. So for instance, if you just want to randomly check for a variant in KRAS codon 12, where there can be different variants that are all pathogenic, you can just type in G12G and you'll see any variant that occurred at that location. If you know exactly which variant you're looking for, then you can always do that. So in this case, we have a C to T here at this position. Okay, so this is how we can see a missense variant. This C in the reference sequence down here is changed with a T. And if we look at here in the coverage track that I'll show you soon, you can see that it appears to be in roughly half of our reads here. Okay, and this CAC that turned into CAT is a synonymous variant. Both codons translate into histidine. Okay, so going on with the general IGV screen here, you can zoom in and out if you want, and then we're going to look at this first track here at the top of the screen. So this has 3 parts. The first is the chromosome band marker. Okay, so this is the chromosome we're looking at. I believe it's chromosome 3. These are the different cytobands on it. The P, the small arm, or Q, the long arm. And this red line here marks exactly where we're looking on the chromosome. So again, useful if you want to know where your gene is located on the specific chromosome. Then we have a scale telling us how many base pairs we're seeing in our window, and the chromosomal coordinates of the position we're looking at on the chromosome. Now I'm going to skip and go to the bottom tracks here. This is the reference tracks. And it kind of looks different depending on how zoomed in you are. So if we look at kind of the middle of our magnification options, we can look at a level that we can see most of the gene. Right? Where there is a gene on the chromosome, you're going to see these blue lines. In the intergenic regions that are not encoding for any RNA, you're just going to see a blank space. Okay, so this is an intergenic region, and there's a gene from each side of it. If we look at the specific gene, I think we already mentioned this. When it's a flat line, these are the introns. When you see a rectangle, these are the exons. And when the rectangle is shorter, these are the untranslated regions of the mRNA. Now, if we look a little bit closer, we can see these different colors that represent the different bases in the reference sequence. And on the exons, we can see the amino acid acids in one-letter codes. Okay? This is a collapsed view of this reference track, but if we open them up and expand them, we can get a little bit more information. So if we zoom a little bit closer, we're going to get the reference sequence. Each base is also color-coded. Underneath that, we have all the three reading frames. And I remind you, if we look at the sequence from the other reading direction, we're going to get three other reading frames. Then, if we expand this view, we can see the different accessions that are available for this specific gene. And again, to show you the importance of knowing your excision number. So we're looking at this R in position 841 if you use this reference sequence. But if you look at the same position in a different reference sequence, the amino acid coding number is going to change. Okay, so even though we're looking at the same exon in these two versions, the amino acid coding number is different. So that's why it's important to know which excision number we're using to make sure that we're looking at the correct reference. Now, if we click on this arrow where the sequence is, we can get the opposite reading direction. Okay, so if our gene is transcribed from left to right, we want to get the reference sequence left to right as well. But if we have a gene that is read in the opposite direction, we're going to want to change the reference sequence to fit our gene. And it's not only the reading direction that changes, you also see the complementary sequence on it. So we're looking at exactly the same location here, but the reference sequence changed when we changed the direction we're looking at. So G changed into C, A changed into T, and so on. So I finished showing you all everything around our sequence, and now I'm going to talk a little bit about the main window here. And this contains two tracks. The coverage track, okay, that shows you how many reads you have for that specific region. So if we're looking at the PDGFR8 gene, each time there's a peak in the coverage here, it means we have reads in that area. And of course, the peaks in the coverage, when you're looking at an assay that is supposed to cover the exons, they will align with the exons in the gene. Now, I want to ask you a question. So please answer in the chat if you can. These are two views in IGV. And if you look at the reads here, which one of them represents a hybridization capture-based assay? So I have a few answers. Seems like most of the people are answering B. And if we look at the answer, it is indeed B. So how did we know that? I want to remind you that an amplicon-based assay uses primers for both sides of the sequence we're interested in. So your sequences are always going to start and end in the same position, where the primer was designed. Okay, so you're going to have multiple sequences. They may have overlap between them, but they're always going to start and end at the same position. While a hybridization capture-based assay uses probes, a single probe, to target the region of interest, and it can capture different sequences that start and end in different places. Okay, so I'll go back to show you what we have. Here in A, we have multiple sequences, but they all begin and end at the same exact position. While as in B, each sequence starts and ends in a different location. So this A here is an amplicon-based assay, and B is a hybridization capture-based assay. And this is one example of a variant we found. So if we start looking at the coverage track, you can see that most of this has a coverage of about 1,200 reads. But here in the middle, there's a gap with a downfall of coverage to about 360 reads and a deletion of almost 800 reads, okay? Now, a deletion, if we look at the red, at the reads themselves, will be seen as a black line, and you'll get a number of the bases that are deleted. So in this case, we have a deletion of 6 bases. Another thing I should mention is that in some views of IGV, you're going to get blue and red sequences. You can customize that for yourself in most versions of IGV. And this means a different direction of read. Okay, so the blue is reads that are from right to left, and red is reads that are from left to right. And it's important to know that because when we get a variant, we want to know that it was read in both directions. Okay? If you get a variant only in one reading direction, that might be an artifact. So in this case, I can see blue reads with the deletion and red reads with the deletion. So I'm good with the quality of this variant. And now let's look how it looks like in IGV compared to the name of the variant. So we're looking at the PDGFRA gene. This is the excision number, and this reference fits this excision number. And we have a deletion of 6 base pairs starting at position 2522 and ending at position 2527. And this causes a deletion of 2 bases, of 2 amino acids, sorry, in codons 841 and 842. The deletion is of GAGACA. But if I look here in the IGV representation of it, I see a deletion of CAGAGA. So it's not quite the same. But if I continue looking at the sequence, there's another CA at the end. So it really doesn't matter whether this CA is deleted or this CA is deleted. The result is going to be the same. There's going to be only one CA left, and the GAGA will be deleted as well. So even though these 2 came from the same pipeline, and the representation is different, but it actually means the same. Okay? Now, the result of this deletion is a deletion of 2 bases. So we have a deletion of this arginine here and the aspartate here. And this alanine, the C from it was deleted, but then instead of this C, we have this C in the sequence, and this amino acid does not change at all. It's still GCC, so there's no change here in this amino acid, and there won't be a change in the isoleucine after it. So it's just a deletion of these 2 base pairs. And just to complete the picture, this was a congenital brainstem tumor that was diagnosed as a diffuse pediatric high-grade glioma with this PDGFRA variant, and methylation analysis confirmed this diagnosis. Now to get back to grounds that we are more comfortable with, this is a sequencing result from a 65-year-old female that had a colorectal carcinoma. And our results were these 4 variants. So first I'd like to ask you which you think are clinically relevant. There can be more than one that is clinically relevant. Please take the time to look at the full results, not only at the gene, at the coding and at the additional information as well. So while you're looking at it, I'll just say that this column here has the clinical annotations in it from ClinVar, Cosmic, that both classify variants, and Nomad, that is a population-based database, just showing variation in the general population. And the column at the end shows the frequency of the variant and the number of reads. So if you had to choose, which of these variants would you choose to report? So it seems that most people think about B and or D. A few are interested in C. No one interested in A. Okay, so let's see how it looked like and see if it changes your decisions. Okay, so I'm going to start with the first variant, and this is the PIK3CA variant. It's on codon 545, and this is the location of the variant. And this is how it looks like in IGV. So after seeing this, wondering if anyone now thinks this is a variant that should be reported. Seems like no one thinks this should be reported. So I saw this also, and I was kind of curious because I couldn't see anything. And then I remembered that PIK3CA has a pseudogene. So I wanted to go and look at that area. So I'm going to show you a more advanced tool you can use to do that in IGV, and that's called BLAT. So what I did is I copied the read sequence. Okay, if you just right-click on any sequence in IGV, this window pops up and you can copy the read sequence. Then you can go into the tools and choose BLAT, copy the sequence into this BLAT window, and you get a result. So BLAT is a BLAST-like alignment tool. That's what it stands for. BLAST is a tool that compares sequences you give it to reference genomes and finds the most fit reference for it. And BLAT does the same thing, but only in your specific reference. So if where a reference is the human genome, it will search for the same sequence in the whole human genome. And when I searched for this sequence, it came up with two locations that had the same score and the same number of matching reads. One is on chromosome 3, and that's the original location of the PIK3CA gene. And the second one is on chromosome 22, and they both got the exact same score. So indeed, PIK3CA has a pseudogene, and it is located on chromosome 22. And then I looked at the reference of this pseudogene. Okay, so our reference, the regular PIK3CA, or the actually encoding PIK3CA gene, has an A in position 1634. Okay, and that's the position that we're looking at. But if you look at the reference of the pseudogene on chromosome 22, that has a C in the exact same position. So most of the sequence will be identical, but just this position has changed. So that is kind of suspicious, but our change was actually A to G and not A to C. So this still doesn't explain explain our results here. So what I did is to go back to our reference, and I changed it into a different mode, that is squished mode. And here you still do not see any of these alterations. Okay, I would expect to see a change in color in one of these. But when I clicked on the coverage track here, I actually did see that change at 16%, and that was what was reported. So I consulted with our bioinformatician and he confirmed that this looks like a real variant. I do not have an explanation why it doesn't appear in my IGV window. It did appear in his. So I'm just saying this to remind you that if you don't see it on a BAM file, but it does come up on your results, then you still need to kind of investigate why that happened. And it doesn't necessarily mean that this variant is not a real variant. So that was this variant. And now I'm going to go on to the second variant. It's a variant in NRAS, and it seems like most people thought this was a real variant. So codon 12 on NRAS is one of the hotspots in all RAS genes. And if you look at this variant in IGV, then you can see the T that replaces the C here in this position. And it's seen in good coverage, right? There's 50% roughly in each of them. So the allelic frequency will be around 50%. This seems like a true variant, but it's still confusing because our variant caller called it a G to A variant, and here we can see a C to T. So if we zoom out, we can see that the NRAS gene is actually encoded on the antisense sequence, and here it's from right to left. So in order to get the correct reading, we need to change the direction of our reference sequence. So if in our first reference, if we saw an ACC, the correct codon is actually a GGT, and our alternative allele, instead of being ATC, is actually a GAT, and these correspond to the correct amino acid codes for this variant. So it's a glycine that had a missense variant into an aspartate. Now this is the view of our third variant in MSH6. And this is the position, okay, of this change. And you can see a few deletions here. After looking at this, does anyone think, what do you think? Does anyone think this is a true variant? So if we look at this, there are two things we should confirm for this kind of deletions. The first is the frequency of this deletion, and here it's only 15 reads out of 448 reads, so that's not much. That still doesn't mean it's not a true variant. It still can be a true variant, but we also want to look at the reference sequence. And in this case, the reference sequence is a homopolymer. Okay, so this homopolymer consists of 8 copies of C. Okay, so we have a combination here of a low variant allelic frequency and a homopolymer region. This would be suspected as an artifact because these are common sequencing artifacts when there are multiple numbers of the same base here. The sequencers might miss one of them, and this and this would be a common variant. And if it is indeed a common variant, you're going to see it in many of your cases and not just in one. Okay, so if you kind of start looking at these results and you see one of these variants that come up frequently, it's probably a sequencing artifact. Now, someone said mismatch repair. That's also correct. If you have mismatch repair deficiency, you can have variations in these kinds of homopolymers. So depending on your clinical suspicion and exactly which gene you saw it in and the potential clinical significance of it, you can try and test in a different method. If you have immunohistochemistry that's available, and in this case it is for MSH6, you can do that. If you have Sanger sequencing that's available, that can also get rid of these homopolymer artifacts. So if there's clinical significance in it, you can consider doing that. If not, I would recommend not to report this kind of variants when they're not confirmed, because they are very likely to be artifacts. Okay. So that was our third variant. And then we have our fourth variant that was a duplication in CREB 3L1. So this is how insertions or duplications look like in IGV. There's this purple eye in the between the bases where the insertion happened. And if you click on it, you're going to see how many bases and which bases were inserted. Okay, so in this case, there's an insertion right at the splice junction of this exon here. And it was interpreted as an insertion to the exon that will cause a frame shift and a termination of the gene after 156 amino acids. Okay. So after looking at this, a few people did say that they think this is a real variant. Do you still think that? Does anyone change their opinions? What would you vote? Okay, so we have we have a couple of people that think this is real. So let's go back and look at the results we received. So it's an insertion in CREB 301. It's actually, it was called intronic because it's right at the junction. And it was reported as a frame shift in this gene. Okay? But look at the annotations here. So we have a nomad annotation, which gives us the population frequency of this variant. And here it's 99.99% of the frequency. Okay, so this is a very common variant in the general population. And in our case, this was present in 100% of our reads. Now, let me remind you that the NRAS was present in 50% of the reads. That can give you an idea of the tumor cell content in this case. Okay, so this is a very common polymorphism. I would even say that this is actually the true reference. If it's present in 99.99% of the people, this should be the reference and not the actual reference. So this variant is a polymorphism and should not be reported. So to summarize our case, this was a case of a 65-year-old female with a colorectal carcinoma, and our results gave us these four variants, but actually only the first two seem like true variants that should be reported. Of course, these we still did not look at the clinical significance of these variants, and if they are indeed clinically significant, they should be reported. I'm going to go on with my third case. This was a 6-year-old male with a diffuse anaplastic Wilms tumor, stage 1, and this is just a picture from the internet to show this type of tumor. And these are the sequencing results. So there's a variant in TB53 that seems pretty much okay, and I want to focus on this variant in APC. Okay, so it's a deletion of a T that results in a frame shift in the APC protein. APC is a tumor suppressor, so a frame shift in it would probably be an oncogenic variant. But when I look at the allele frequency, it's very confusing. Okay, so there are different alleles here, and each one has a different allelic frequency, and I do not understand it from this view. So I went to IGV to look at it. and it actually looked like here. Okay, so this is the position that we're interested. This is the T that was deleted. But it actually seems like there are a few deletions here, right, with the black line. But most of these alleles are a missense variant where the T turned into an A. So actually, this is not a frame shift deletion. This is actually a missense variant, okay, where isoleucine in this position changed into lysine, okay? So if we look at it closely, at both sides of this T, there are A's, okay? So 3 A's on this side and then 4 A's on this side. And this missense variant changed the T here into an A. And that resulted in a homopolymer sequence, right? Just like we saw before. So this, in some of these reads, the sequencer missed some of the A's, okay? And because the T here was absent, it was read as a deletion of the T, while in fact it should have been read as a missense variant in this position. Now, just to see if this is a known variant, we looked for it in IGV, in ClinVar, that is a population-based database of genomic variants, and it is reported in ClinVar. It has different interpretations. The most common one is as a risk factor, but it also has interpretations of pathogenic or likely pathogenic variants, or as a variant of uncertain significance. So to go on, we looked at the general population, and this is a fairly common variant. 3.6% of the general population is pretty high, and it's especially in one population, in the Ashkenazi Jewish population, and this child belonged to that population. And you can see that there are multiple alleles that are reported with this variant, and some of them are even homozygotes. So if it was found in homozygotes, it's probably not an oncogenic variant, but it is considered as a risk factor for different types of cancer. So it actually might have some clinical significance, and it should be reported, but it should be reported accurately, and using IGV in this case helped us report it correctly. So now I'm going to go on to my fourth and last case for today. And this is a 53-year-old male with a metastatic medullary thyroid carcinoma. He was diagnosed and the tumor was excised 8 years prior to testing. At that time, sequencing was not available for thyroid carcinoma, but it became available, and the oncologist asked for testing of this tumor. The patient was still metastatic, but there was no new tissue that was available for testing, and they couldn't biopsy a new lesion. So we had to try using the old biopsy from 8 years ago, and we talked about it a little bit last week, and specimens that are older than 7 years are not recommended for sequencing, but in this case, we didn't have a choice, so we did it anyway. And this was sequenced in a comprehensive panel that gives you all sorts of results. And one of the results it gives you is a TMB, a tumor mutational burden. And in this case, it was 500, which is extremely high tumor mutation burden. And these results are after a filter. So it's not all the results were found in this specimen, but it still had 1,700 variants that were detected. But if you look at it closely, it seems that most of these are C to T or G to A, which is essentially the same thing. Okay, so C to T or G to A variants. most of these with a fairly low allelic frequency. Okay? The tumor cell percentage here was estimated to be 80%, and all of these variants are present in less than 10%. It seems like most of these variants are actually deamination artifacts. So deamination artifacts usually are mostly C to T or G to A, okay? And adequate fixation can help minimize them. But when you have an old specimen, like in this case, that won't help. You can do a pre-treatment with uracil DNA glycosylase. And what the reason this works is that the deamination actually changes the cytosine residues into uracil. Uracil in RNA is the RNA parallel to the T in DNA. Okay, so when it's sequenced, it's read as a T. And what this pre-treatment does, it just breaks the DNA strands when there is a uracil residue in them. And it gets rid of most of these. But if you have a very old specimen and you use it, you will probably break most of your DNA fragments, and you'll end up without sufficient DNA for testing. Okay, so we basically need to be aware of this type of variants. And the importance of it is that if you get multiple deamination variants, you can't really tell if you have a true variant that is from the same type, right? A C to T or G to A. So you need to be especially careful around these. But if you have a specimen with a high enough tumor cell content, you might be able to see these, the true variants, if you filter it correctly. You should always check the BAM file and look at your variants. And when it's possible, and you see a variant that is real, if you're able to test with other methods to confirm, that would be the best option. So going back to our case, we customed a deamination filter that basically got rid of all the variants with low allelic frequency, and we made it higher and higher until we were left with a reasonable amount of variants that we could look into more easily. And this was the list of the variants that we saw. And one variant popped up here, and that's this variant in the RET gene. RET mutations are associated with medullary thyroid carcinomas. So we had a look at this variant in IGV. Now, let's just look at it. So this is a deletion of G and C and insertion of TT. So there's no change in the number of bases we have. It's just a change of GC to TT, which caused a missense variant. And this is how it looked like in IGV. Okay, so we have these G and C that changed into TT. And it looks like a pretty high allelic frequency. If we click on the coverage here, we can see the allelic frequency of it, and T here in the where the C was in the reference, T was present in 30% of our reads. Since in this case we have an alteration of 2 base pairs, if we want to confirm that it's real, it's helpful to look at where they are. So in all of these, they always come together. It's always GC that turns into TT. If this was spontaneous deamination of this C, for instance, then we would not see it in exactly all the reads that have this that had the G to T alteration. We might see some that have both, but others would have only the G to T, and others will have only the C to T. But here, they always go together, so this confirms that this is a real variant. And if we search for it in the databases, then it is reported as a pathogenic variant in ClinVar, and it is associated with medullary thyroid carcinoma if you look in Cosmic. So that fits clinically as well. This panel that was used also has an option to visualize which mutations you have and the pattern of somatic mutations when you have multiple mutations. So in this case, more than 75% of the variants were C to T, the type of mutations that are seen in deamination. And this also confirms our suspicion that there was a lot of deamination artifact here, and it's not a real high TMB. So we could report the RET mutation in this case, but we did not report anything else, and we gave a comment that the quality of the specimen was inadequate to report anything else. So to summarize what we did today, we looked at the bioinformatics pipeline, which is the process that starts from the sequencing results and ends in a variant caller file. We started from the raw data, then that was the result of the sequencing. Then we saw that there was a base calling process that resulted in an output file that's called a FASTQ file that contains the name of the file, the sequence, and quality data about the sequence. This file is then aligned against a reference file to result in an output of a BAM file. The BAM file can be viewed in IGV, and we showed a little bit how to interpret the BAM files. But still, BAM files are large, and you can't scroll through the whole BAM file to identify your variant, so it needs to undergo a process that is called variant calling that identifies the variation between your sequence files and the reference files, and the result of this is a VCF file with a list of your variants. But the VCF file itself is, again, not enough because we do not know these locations, that they don't mean anything to us. So this file undergoes a process that is called annotation, where these coordinates are translated into a gene and a protein change, and data from various databases can be added to it until we get a annotated variants file. And this is where we end today, but this annotated variant file may contain thousands of results. Some of them might be false positive variants like sequencing artifacts or deamination artifacts, or just random variants that are in a very low allele frequency and not relevant for to be a driver in the tumor. Other variants might be true variants, but without any clinical significance, like single nucleotide polymorphisms, or variants that are benign or likely benign. So these type of files, the annotated variants files, still require filtering. And these are just a few suggestions on how to filter them. Of course, most pipelines do it in some way. So we want to look at variants that have an allelic frequency that is between 5 and 95%. Anything lower than 5, usually in most clinical assays, is not accurate, and over 95%, it might be just a polymorphism that is present in all of the person's cells, and it's not relevant for cancer. We want to look at the population frequency. We want to filter out known artifacts, and most importantly, we want to know the clinical significance of these variants. And that's achieved by knowing our variant classification schemes, which we will talk about next time. So with that, I'm going to end today's session, and if anyone has any questions, I'd be happy to answer.
Yeah, that was a great talk, Dr. Fisher. So thanks for giving that. And please join us next week for the final session in this lecture series, which will be about interpretation of these molecular results and classification. And make sure you fill out your evaluations. So we'll stick around for another couple of minutes, I guess, in case people think of any questions. But people seem to have really enjoyed that lecture.
Thank you all for joining us.